Oh,
Just started.
excellent. Okay, so as I was talking with Dan, actually, I'm Yiannis Koutalos, and we can keep it informal. So shout. I don't know if raising a hand actually, an electronic
can
hand
interrupt,
actually
I
would work.
think.
Yes, feel free to interrupt. Let's put it this way. And so this is actually the measurement of NADPH generation, and I would be describing this work. This has been going on for many years. So these are the people who have actually done the work, Chun-He Chen and Lee Adler in my lab. And these are most of the results that I will be presenting is from these two papers, actually. The first one, which was mostly in the mouse cells, actually, is from 10 years ago. But there is actually a very recent one that was actually, we did this work in a human, and we can talk why it took us that long actually to finish this. But we are actually done with that part with human cells as well. Anyways, and the funding actually, it's important to mention, is from National Eye Institute and also from the Barbara and Stanley Andre Endowment. So without further ado, I will start discussing the mouse situation, NADPH generation in mouse rod photoreceptors. This is the cross-section of the mouse retina, and the sclera, the white part of the eye, and the photoreceptor cells, the ones that we'll be dealing with, are here next to the retinal pigment epithelium. And these are the outer segments of the photoreceptors and the inner segments. Outer segments, the business end of the cell, where transduction of light to an electrical signal takes place, and inner segments where the metabolic machinery is located. And this way of presenting it, the light is coming from the bottom here, actually reaching the outer segments. So the reaction that we'll be dealing with is actually part of the visual cycle. And this is actually something that in the retina, this is the retinal pigment epithelium. These are the photoreceptor cells here. And this is the outer segment. Outer segment contains the visual pigment, rhodopsin. It has 11 cis, the 11 cis isomer of the chromophore, of the retinal chromophore, as the light detecting part. And what light does is isomerizes the 11 cis chromophore to all trans. And this generates the active intermediate through which we get light transduction. This photoisomerization, which is the first part of light detection, destroys the pigment. And this actually necessitates the regeneration of the pigment. Otherwise, we would not be able to see. And we're all familiar with this, right? If we go from a bright environment to a dimly lit environment, it takes a while to get our sensitivity. And this is the reason for this, is that this needs to be regenerated because the bright light has destroyed a lot of it. And the first step actually in the regeneration is the release of all transretinal. And this takes place in the outer segment and then leaves behind the apoprotein opsin. And then opsin is going to combine with fresh 11 cis retinal that comes from the retinal pigment epithelium to actually regenerate rhodopsin. I will not be discussing any other part of the visual cycle today, but this release of all transretinal actually provides us the means, if you like, to look at NADPH. And the reason for this is that all transretinal is reduced to all transretinol within the outer segment. And this reaction is catalyzed by RDH8, retinol dehydrogenase 8, and uses NADPH as a cofactor. And there is actually, with bright light, there is a lot of all transretinal being released and a lot of all transretinol being formed. And the metabolic machinery of the cell has to keep up with this. So has to keep providing NADPH. And we will see actually that it actually does that. And so this is basically the reaction within photoreceptors that I'll be using, we have used actually to look at NADPH generation. And the reason we can do that is that because these two guys fluoresce. So it's a fluorescence assay, basically. So we work with isolated rod photoreceptor cells. This is from the mouse. And these are the photoreceptor cells here. And we chop the retina. And this is something that we might want to talk about later for the isolation procedure. And we get isolated cells. This is the outer segment of the cell, the inner segment, and you can see still the nucleus. We do lose parts of the cell, and that is actually relevant. But the metabolic machinery and the outer segment are there. So, as I said, we will be using fluorescence imaging, and the way that the assay works actually for that is that outer segment, these are the reactions that take place in the outer segment. We'll start with dark adapted cells. We'll actually shine bright light, isomerize, then all transretinal is being released. This reaction is going to be taking place. And the NADPH that is being used is provided by the metabolic machinery in the inner segment. And as all transretinal is released, is reduced, more NADPH has to come in to keep this reduction going. And for fluorescence, we actually chop the retina. We get isolated cells. This is a mouse cell sitting in the bottom, in a chamber, and at the bottom of the chamber, actually, in a microscope. And then we get a light, we have a light source. We select a particular excitation light, reflected by the dichroic mirror, focused by the objective lens of the cell. The cell emits fluorescence. The lens collects the fluorescence, and the emitted light goes through the dichroic mirror. We select particular wavelengths that we might be interested in, and goes on to a high sensitivity CCD camera, and you can actually see the fluorescence image of the cell here. And this particular one is actually retinoids that you see. Now, this is basically what you see. This is a true color image. So this is actually the Nomarsky image of the cell. And this is when you excite with 360 nanometers with DAPI optics, essentially. But if you look through the eyepieces and you're fast enough, this is what you're going to see. And this was actually caught with a color camera. And this fluorescence in the outer segments is from all transretinol and all transretinal. This is retinoids. This one is NADH, mitochondrial NADH. And you can see that this color difference that you see is actually real. There is actually a slight difference in the emission spectrum between NADH and the retinoids. And you can see it actually with your eyes. And we have measured spectra and so on. So these are actually retinoids, and they have very similar emission spectra, retinol and retinal. So to actually be able to do the assay to look at the conversion of retinal to retinol, which you need for the NADPH measurement, we need to be able to distinguish. We can actually do that. And the reason for that is that because retinol and retinal have different excitation spectra. So this is actually in a cuvette, these are absorption spectra. They're very similar to excitation spectra in this case. And this is all transretinol peaks around 325 nanometers, all transretinal 380 nanometers, and there is an isosbestic point around 340 nanometers. So we can actually, by using different wavelengths of excitation light, we can distinguish between these two species, these two retinoids. And this is showing you that we can indeed do that. These are broken off rod outer segments, and they do not have any connection to metabolic machinery. They're relatively inert, pretty inert, and we load them with a lot of retinol or retinal, and we excite at 340 and at 380 nanometers, and we collect the emission above 420 nanometers. So 340 is here, so for retinol, 340 over 380 down here, it's going to give us much higher fluorescence at 340 than at 380. Retinal is going to be the other way around. 340 is going to be less than 380. And you can see that indeed, actually, you can get in terms of the fluorescence signal, we can get retinol much higher 340 than 380. Retinal is the other way around. What is important here is that we can actually go and measure the intensities and measure the ratios of excitation by 340 over the excitation by 380. And this is actually shown here. This is the ratio of 340 over 380. This is the ratio for retinal. This is the ratio for retinol. And if you have done any fluorescence imaging with calcium-sensitive fluorescent dyes or pH-sensitive, whatever, this is basically, it's a ratiometric assay. And we have actually just worked through it to actually use it for the retinal-retinol ratio. And I'll be going through this in a moment. So, but basically the idea here is that we can use the 340-380 ratio excitation rate to measure the conversion of retinal to retinol, how much of each we have, and this actually would reflect NADPH generation. And basically this is the Fura set, and if anybody has done calcium imaging, the filter set that we are using is the Fura set. Okay. So does this work? And yes, it does. So this is actually an intact, and by intact I mean metabolically intact rod photoreceptor, isolated. This is the outer segment. This is the inner segment with the metabolic machinery. And this is a broken off rod outer segment, has no connection to metabolic machinery. These are dark adapted cells. So they start with a full complement of rhodopsin. And then what we do, we measure, take fluorescence images, 340 and 380 nanometer excitation, right? These are here. The dark state for the intact and the broken off. And then we shine a lot of light for 1 minute. We bleach rhodopsin. We bleach about 95%, 95% from the way we have measured it with biochemically, actually, just when you do the same under the same conditions with rod outer segment membranes, this is how much we bleach. And what you see after 10 minutes after light, where actually retinal has been released and quite a bit has been converted to retinol, you can see actually you have much higher fluorescence when you excite at 340 than at 380, goes on until 60 minutes. When you look at the broken off outer segment, it's the other way around. You do not have reduction. And these are the reactions, right? All transretinal going to all transretinol if you have an NADPH. If you do not have an NADPH, you stop here, as in this case of the broken off.
What's the medium that's bathing these?
So this is actually a physiological solution that contains 5 millimolar glucose. So this is an important question, and it's something that we could discuss at the end. It took us a lot of time to figure out what is a good solution actually to keep the cells happy. It does not have bicarbonate. So that could be a relevant question. We have done experiments with bicarbonate with whole retinas. We have not seen an effect of bicarbonate on the conversion of retinal to retinol with whole retinas. But it is actually no bicarbonate, but has 25 millimolar hippies. So it is very strongly buffered. And when we did experiments with bicarbonate with whole retinas, we actually had the 25 millimolar hippies as well. And the selection of hippies is, one is easy, but the other thing is that these are the 37 degrees centigrade, and the pK of hippies at 37 is 7.4, which is actually the pH that we want to have things at. Okay. The composition of the medium is published, but and this is something that we could actually touch upon later, because it works. We are happy it works. We do not know how and why. We have tried different ways, and this is the way that it worked. We took a recipe for a culture medium with 25 millimolar hippies, and it worked. So when you actually plot the ratios, this is what you get. This is the 340 over 380 ratio here as a function of time after bleaching of the cells that start as dark adapted. And this is the intact, and this is the broken off outer segment. This is the ratio for the retinol that we got from the beginning, right, with the broken off outer segments when we loaded with retinol and retinal. This is the ratio for retinal. So this is going to be the max, this is going to be the minimum. And you can see that the broken off outer segment is basically retinal. We cannot distinguish it from retinal. So what is retinal is being released and it stays retinal. In the intact situation, we are getting close to retinol, not fully, but it actually have plenty of conversion to retinol. Now, what we can do now, we can actually go and run the numbers to see actually, can we get the fraction of retinal that has been converted to retinol? And this is basically what you do with ratiometric imaging. We have done the same thing. What is important for the math here is actually that we are using 340 nanometers, and the 340 is the isosbestic point, and that helps quite a bit. So this 1.8 and 0.14 come from the ratios, the maximum and the minimum actually. This is the actual ratio that you're measuring. And Q here is the quantum yield ratio of retinol over retinal. And that we have measured independently. And we can talk about it. We've done it in a couple of ways, and this is the range that we have gotten. But this allows us actually to use the ratio to convert, or to obtain from the ratio to obtain this fraction of retinol over the total chromophore that is being released. Retinal is being released, some of it is converted to retinol. This is the total chromophore that has been released, and this is the fraction of it that is retinol. Now we can actually go ahead and use this ratio along with the reaction rate that converts retinal to retinol that uses NADPH. It uses also protons. We know the equilibrium constant of the reaction from a very old paper by Bliss. And the assumption here, in order to be able to do the calculations, is that the reaction is at equilibrium. And on the basis of the ratio being relatively stable, actually, over time, it seems actually that this is a reasonable assumption. And then we can actually run the numbers again. And this is basically what you could get for the NADPH fraction. When you actually put this fraction here over in this equation along with the definition for the equilibrium constant, this is basically what you get. You do need the proton concentration. So one needs to go ahead and measure the outer segment pH. Now, a couple of things that are important here is that these numbers here, and they do not depend on any assumptions about the NADPH or the reaction or anything like this. This is basically straightforward doing the ratiometric imaging. The fraction of retinol that we get is about 0.8. And that's actually is in agreement with our biochemical measurements, HPLC measurements, from whole retinas. And I can show you the data at the end if there is interest. The other thing is that when we plug in the numbers, we'll go over it in the next few slides, with protons and everything, the numbers that we get for NADPH fraction are in agreement with histochemistry. And we'll get to that in a moment. So, first, we need to measure intracellular pH in order to actually go from retinol fractions to NADPH fractions. And this is starting, we use BCECF. We use the acetoxymethyl ester form. Very nice dye, actually. Very consistent and very bright, very straightforward to use. And it's a ratiometric dyes, 440, 495. And this is a mouse rod photoreceptor loaded with BCECF. This is what we are interested in, the outer segment fluorescence. There's plenty of dye in the inner segment. The acetoxymethyl ester is cleaved, the dye is trapped. And we can actually start with a dark adapted cell, and we measure the fluorescence initially, and we can get a ratio for that. Then we bleach for 1 minute, exactly the same experiment that we are doing for the retinoid measurements. And then we look at pH over time. And you can see actually that pH starts about 7.3 in the dark and goes down to about 7.1, about pH 7 over time. And so the cell acidifies. And
do you have the same measurements for the cytosol, or for, I guess, the inner segment?
This is cytosol. We haven't done this, and I'm uncomfortable doing that for the inner segment. And the reason for this is that I don't think one can tell, especially with epifluorescence, and even with regular confocal, I don't think you can tell what is in the inner segment cytosol, what is in the mitochondria, what's in the ER. Really, everything comes together. So the dye is going to go everywhere. It's not going to stay just in the cytosol in the inner segment. It's going to go into mitochondria and the ER as well. So it's going to go into mitochondria and be cleaved in the mitochondria as well. So I don't think we can distinguish. I haven't tried to do it. We have the data. It is not saturated. In the actual data, it's not saturated. I've actually cranked up the gain so that you can see we have plenty of fluorescence in the outer segment to actually do the measurement. And there is no related fluorescence sufficiently bright in the outer segment that could interfere with this. This is a clean measurement actually of pH. So what you see is BCCF basically. So what we have been using actually as the outer segment pH for our measurements is 7 to do the conversions. So we do this. So this is what I showed you before. This is the measurement. This is the ratio actually. And when we plug in the numbers for the ratio and for the pH, we get actually what the fraction of NADPH is. Retinol is going to be 100%, of course. Retinal is going to be 0. And this is about 20%. We have about, in the outer segment, NADP is about 20% reduced. And this is actually in agreement with histochemistry. This is what I was referring to before. Back in 1968, Machinsky actually had done some very, very nice work for rabbit and monkey and retina sections, and did the histochemistry to measure the levels of NADPH, NAD, NAD+, NADP+, and so on and so forth. His numbers were about 30% for the outer segment layer. He measured different layers. So we're broadly in agreement, actually. And that's something to discuss again, whether these numbers make sense. That basically the idea here is that for reactions that can go either way, you need something that is not fully reduced or fully oxidized. For glycolysis, NAD is fully oxidized, essentially. You have NAD+. So it's a driver. This one, NADPH, is not going to be the driver of the reaction, basically. That's the idea behind it. At least that's my interpretation of these numbers here, that, you know, they're good numbers, that the reactions can go either way. Okay. So just to remind you, because now that we have an assay, we want actually to see, okay, what can we use it for? It's great, but can we do something interesting with it? And so this is actually the pathways for generating NADPH. The typical one that we learn in biochemistry is the pentose phosphate pathway that you get from a branch of glycolysis. You get NADPH in this way. However, there are actually other pathways that involve the mitochondria, and it's isocitrate dehydrogenase that uses isocitrate that comes from the mitochondria through citrate, and also malic enzyme, malate, and you could have malate dehydrogenase that would actually generate NADPH as well. So we can have glycolysis pathways, the pentose phosphate pathway, and also mitochondria-linked pathways. So we got quite excited about it, and we decided to check, is NADPH coming from the pentose phosphate pathway in mouse cells? And that's what we learn, right? Is that NADPH is coming from the pentose phosphate pathway, typically. And what we did actually, we went ahead. So we did the experiment with glucose, 5 millimolar glucose, right? This is dark state, bleach, and you can see the formation of retinol. Then we actually went to zero substrate, remove the substrate, and this is tricky, and we can get to this. But this is basically the result that you get. You do not have you have reduction, but you have plenty of retinal left over. And then we actually said, okay, this is very promising. This is not exactly completely retinal, and this is an important issue. And but if we actually use glutamine, we get very nice reduction of retinal to retinol. So this is an important point here, that if we go to zero substrate, we basically knock almost everything out. Almost, but it's good enough. Otherwise, if there is actually sufficient intracellular stores that could keep this going, we would not be able to do the rest of the experiments. So this is an important point, that what this shows actually is that the mouse actually does not have, mouse rod photoreceptors do not have high levels of intracellular stores. This allows us to do the experiments that you will be seeing. And that's not necessarily the case for all photoreceptors. So that's something to keep in mind. So this is basically what one gets, and can actually do the conversions to NADPH. This is glucose, 5 millimolar glucose. This is 0.2 millimolar glutamine, very nice. Basically the same as glucose at high concentrations. And 0.2 millimolar glutamine is pretty good, close to what you expect physiologically. 5 millimolar glucose, what you expect physiologically. Glucose, 0.5 millimolar, 0.2 millimolar. These are reasonable numbers. And in the absence of a metabolic substrate, you get a pedestal here. There's something extra. And I interpret this to be due to intercellular metabolites. So there is something left over that actually can sustain the reduction, can sustain the generation of NADPH to keep things going for a while at least. We haven't done this in the mouse. You will see data in the human in a bit to actually test whether this is indeed intracellular stores. We just never got around doing it. But this actually shows that glutamine can actually support NADPH generation in the absence of glucose.
Do you have a sense of whether, I mean, maybe you'll get to this data, and I'm sorry if you will.
Yeah.
Do you have a sense of whether glutamine is sort of being reductively, or the alpha-ketoglutarate glutarate that's coming from glutamine is being reductively carboxylated into isocitrate and then going to citrate, getting exported? You know, it's kind of a complicated path for the IDH stimulated form of NADPH production, right?
So yeah, so this is my interpretation is that what we see from glutamine is actually the malic enzyme and the isocitrate dehydrogenase. I do not know which one. And we'll get to this in a moment. There is a relevant point here that Jim was asking about over the years, because we've tried citrate from the outside and it doesn't work. So, but we'll get to this in a moment. Okay. So can other amino acids be used as substrates? And since we can do glutamine, why not? Mitochondria can pick up other things. Will they be able to use it? And this is an experiment. This is all amino acids to be able to compare. Just to be able to do a comparison. These are not the physiologically relevant concentrations, but it could be even worse with the actual physiologically relevant concentrations. And this is actually a couple of examples. Aspartate, maybe. Alanine, pretty bad. It does not seem to be doing much. And these are actually the results for different amino acids. And what you see actually, glutamine, basically the same as glucose. This is 5 millimolar glucose. Everything else is at 0.5 millimolar, and this is with no substrate available. And maybe there's something glutamate, at least it's significantly higher than in the absence of substrate, and aspartate as well. But every other amino acid was not significantly different. So our interpretation from this is that glutamine is the only amino acid that can support an NADPH generation. We were surprised by glutamate because actually glutamine goes through glutamate to actually get into the participate in the mitochondrial pathways. So keep in mind that these are isolated cells. We are losing components, and it could also be the case that it's simply glutamate goes to a different compartment, and it's not actually used as a substrate for this pathway. So it's not going to actually end up in the same pathway as glutamine. And glutamine, my interpretation is that in the retina, it's going to be coming from glia, from the Miller cells. But this was the take-home message from here is actually that nothing else works as well as glutamine. Taurine does not work either. We tried taurine as well. It does not work. And we also tried malate and citrate. They do not work. Citrate is pretty low. But the other things that we tried, we did titrations. This is a substrate. This is the ratio with glucose and glutamine. And you can see, yes, it does drop off about below 0.1 millimolar. You have the break actually, really you go very low. So this is down to 50 micromolar. At 50 micromolar, really, you lose actually support for NADPH generation. We tried pyruvate and lactate. Lactate because of the old, this is, again, it's a long time ago, so this was the old controversy between Tsakopoulos and Winkler, Barry Winkler. And Tsakopoulos was a friend, and he was wrong. So Barry was right. Lactate does not seem to be doing much, as one might expect. And Tsakopoulos and Poitry had the theory, actually, the notion that lactate is generated by glia, actually, to be provided to the photoreceptor cells to be used as fuel. The other idea was that, well, photoreceptors actually generate lactate, which is what actually seems to be happening. But in our case, at least, we put lactate from the outside. It doesn't do anything. Pyruvate is a little bit better. And this is actually at 2 millimolar or so. We tried malate and citrate at 2 millimolar. Malate was maybe as effective as lactate. Citrate was down here. It was way down. So it was really, citrate did not do anything. So our conclusions basically is that from all this is that NADPH can be generated by mitochondrial-linked pathways. Glutamine can do it, right? And glutamine and glucose are the preferred substrates, and that the NADPH fraction for either substrate is about 20%. Other amino acids are ineffective, and these guys are ineffective as well. The pyruvate seems to be a little bit better.
Did you add these amino acids in the absence of glucose or the presence?
Absence. If you have glucose, all bets are off.
Yeah.
They will use glucose. So, and this is actually the tricky part. Because when we isolate the retinas from the mouse, so this is freshly isolated retinas, they're sitting in 5 millimolar glucose. So to actually wash off glucose, and you saw actually, it's pretty good down to 50 micromolar. And 50 micromolar for 5 millimolar, that's 1%. So even at 1% dilution, we could have plenty of support of NADPH generation. So that's why it's actually tricky to make sure that you actually have removed glucose. So we actually, the experiments that we have done in this way, we actually went to great pains to make sure that we don't have glucose. We check the cells, basically. Do we have anything? When we do not add anything, and the answer was that, no, okay, we're getting the zero glucose effect. So we're quite happy with that. But yes, this is actually a tricky point that one has to make sure that does not have glucose and has to keep washing, making sure actually that you get really diluted away.
Can I make one more point, which is just whenever I've isolated mitochondria in the past, you can't really just add kind of one substrate for continued respiration. You always have to add, pyruvate and malate, pyruvate and glutamate, something like that, because the TCA cycle requires an acetyl-CoA donor and an acceptor to continue to go. And so I guess the question is, do you think that you're not seeing an effect of these individual metabolites because they're in isolation and you don't have a kind of acceptor in the TCA cycle, or because they're just not being transported in? What's your opinion on that?
Yes. My uninformed opinion in this case I had not actually tried to even think in those terms. So I was actually saying, I have an isolated cell. I'll give you what I can do. I can give you good luck. And they were not doing well. So clearly, these are things that one could try. But I had not thought in those terms at all. What I was assuming is that the cell has enough things inside to get started, maybe, hopefully, but maybe they did not have enough.
I think there are just so many anaplerotic pathways that even if it has a TCA cycle, the transaminases might still be working to take some of those carbons away from the TCA cycle, just as an example. So I think this idea of a sort of self-regenerating cycle is like kind of true, but not realistic.
Yes. No, I hear you. So yes, we have not tested any of this, and I think that's an interesting question and an important point. These are isolated cells because in the whole retina, of course, you have all kinds of things coming in as well, right? And you have support from the other cells. These are isolated cells that are really on their own, and so they might be missing something that is actually relevant in this case, and that's a possibility. My interpretation has been that it's transporters. But it could be that it's very well wrong. As I said, it's uninformed opinion.
It could be both. Transporters
It
are
could be both,
super
yes.
important. So
Yes.
Definitely, yeah.
Yeah, so the glutamate was the case actually where I felt that it was a transporter issue or the compartmentation issue. But the other ones, I cannot tell.
Yeah, this is
Okay.
Can
Yes.
I ask a question? This is Jason Miller over at University of Michigan. It's a naive question, but can you put on the glutaminase inhibitors to have confirmed that the pathway was
No,
presumably through that and not something else?
No. Again, maybe I was even more naive than you. I thought that this is okay. Glutamate works. What else? So I was actually happy enough that it was something other than glucose that could do it. And I did not actually try to go ahead and try other things. One of the things that we never did, and it's a sore point actually, we never tried ketone bodies. And this we got sidetracked. So there were tons of experiments to do here. Clearly, it's a robust assay. Actually, it's a pretty robust assay, and you can see it in the human situation as well. You can actually think a little bit more with other things as well. So there are two more types of experiments that could be relevant. What I would like to do is that I would like to move on to the human situation because I think it's interesting. And but then there are a couple of things in terms of the competition between ATP generation and NADPH generation, and also where in 5 millimolar glucose, where does an NADPH come from? Are the mitochondrial pathways actually relevant or not? So I have left this at the very end. We can go over them, but I do want to go over the human data. So at some point, actually, we're able to do living human photoreceptors. So this is actually isolated human retina. And from human donor eyes, these are going to be at least a day after death. So these retinas have been actually subjected to about at least 24 to 36 hours, sometimes 40 hours of ischemia. So the globes are isolated. They put in, are enucleated actually, maybe 5, 6, up to 10 hours after donor death. They put in moist chambers in the dark, shipped to us, and do the dissection and get an isolated retina. You can see the macula here, the arcades. This is where the optic nerve was. And we can isolate single cells, rod photoreceptor, cone photoreceptor. And for the rods, this I will be talking about rods, with one exception, we get them actually from the this ring, the rod rich ring around the macula actually. This is where it is. This is actually the area that we focused for isolating rods, with one exception that we got some from the central area as well. Excuse me. So first, we can do the same type of conversion actually. You can look at the same type of conversion of all retinal to all transretinol. In human cells, this you have seen before. This is a broken off rod outer segments from human, again, retinol, retinal, and these are the ratios in the human case. Slightly different, they're very similar to mouse. This is a little bit less. We just, in terms of rigor, if you like, mouse is about 6.9, this is about 6.2, but retinal was about the same. So we can do the conversion in the human case as well. And this is actually in the cases where we were fortunate enough that the eyes that arrived, the retinas actually dissected, had enough pigment, because people die under bright lights usually, and most of rhodopsin is bleached. And this was typically the case. We could not get any pigment. But in some cases, we were able to get pigment. In some cases, we were also regenerated the photoreceptor cells. But this is with endogenous all transretinal, same as in the mouse. And you can see actually the dark state, 10 minutes after light, 340/380. This is retinol, pretty much retinol actually, 80% retinol. And we do the same thing for the human. And this actually corresponds to an NADPH of around 20%. We measure the pH in the human case. I have the slide somewhere at the end. It's about 7 as well. What you would expect for bleached cells, for bleached cells as in the mouse. So the numbers basically work out to be the same in the human and the mouse. And this is actually important here because these are eyes, retinas that have been subject to ischemia for a very long time. And we get actually same NADPH generation as in the mouse, that is actually, we have an acute isolation. And also compared to monkey as well. And also in that case, we had similar results, and those were not subject to this long ischemia. Now, because we could not get pigment in most of the time, we actually modified the assay a little bit, and we actually used exogenous all transretinal. So we gave all transretinal to the cells from the outside. So this had the pigment bleached. We ensured that the pigment was bleached, so there was nothing, no rhodopsin. There were negligible amounts of rhodopsin left. So we give all transretinal, it will be reduced, and then we can actually do the same, run the assay the same ways. This is the situation, outer segment, inner segment, initial, and we give 5 micromolar all transretinal. And this works well. This is published. This is actually, it's a good concentration. Things work well. From the 340, we can get the level of fluorescence. We can do the same thing with the endogenous retinal. We have actually a measure of how much retinol we have. And this is relevant. So this is the ratio, retinol. This is the ratio that you get for endogenously released retinal, for whatever rhodopsin they have. And this is what you get with exogenous retinal. And the NADPH fraction from this ratio is about 20%. So with exogenous retinal, we get the same 20% NADPH as we would get with endogenous retinal, as we get in the mouse. However, when you look at the amounts of retinol that are being generated, it's much higher with the exogenous. It's 3-4 times as high as with endogenous. So that fluorescence from 340, right, excitation, was much higher with exogenous than with endogenous. And this actually suggests that the human rods have very high capacity to reduce all transretinal. Really, we overload the cells with all transretinal, and they can do very well. They can keep up. They have limits. We go to 10 micromolar all transretinal, they cannot make it. They cannot keep up. So, but basically the capacity is actually several times higher than what is needed for the physiological demand of the retinal to retinol reduction. And we have actually done a glucose titration with the exogenous all transretinal, and very similar to the mouse, breaks up a little bit below 0.2, 0.1. But it's overall it's quite similar actually in terms of the dependence on the extracellular glucose concentration. And this is actually the interesting thing here. When we do not have extracellular substrate present, This is the same as all transretinal. But this is actually plenty of all transretinal. So maybe any intracellular stores cannot keep up with it. And we never tried seriously actually to check whether the endogenous all transretinal would give us the same result as in the mouse, right? And we never did the experiment in the mouse with exogenous all transretinal, overload the mouse. Can we exhaust the intracellular metabolite pool? So we have never done the experiment that would actually decide whether any reduction that we see in the absence of extracellular substrate is due to intracellular pools or maybe something else. But here actually you do not see any difference from all transretinal. So the thing that was of interest in us in the case of the human, in the human situation, was to compare central and peripheral rod photoreceptors. So you saw the retina, we can actually go ahead and isolate rods from the center, from the more or less the macula, but within the ring, the rod-rich ring. And the reason for this is that there is the macular degeneration issue, of course, which is the macula more sensitive to metabolic insults? Is there something special about the cells in the macula? And it has been known actually that rod photoreceptor cells die in the macula with age. There's more dying in the macula with age. Rod cells die in the macula. And in the age-related macular degeneration, actually, things are worse. This is Christine Korsch's work. And this is actually something that drove this project that we wanted to check. And isolating cells actually from the central area is actually difficult. Rods included. Cones, which is the cone-rich area, it was very bad as well. So basically what we have found is that it has been very difficult to isolate. We felt that the macula was not as healthy as the periphery. Maybe it did not have as many cells, maybe it did not have as many rods, but it was very difficult for cones as well. So what we saw actually, that with a lot of cells, there is a significant difference between peripheral and central rods. So there is actually less NADPH generating capacity in central rods compared to peripheral. But in terms of the spread, you can see, is this an intrinsic difference in the cells? My interpretation is that it is not. I think that just the macula is actually more susceptible to the ischemic shock, actually, to the ischemia that the eyes have been exposed to. So, and this is actually just, these cells are sicker, basically. And this is what it shows. And if you wait for a few days, actually, keep the retina for a few days, you will see actually NADPH generating capacity going down. So macula is more sensitive to the ischemic, insult actually than the periphery. That would be my interpretation of this. And with lower cell numbers, unfortunately, when we actually tried 0.2 millimolar glucose, less substrate, oligomycin inhibit ADP synthesis, right? And so push things around in terms of metabolism. And then oligomycin along with monocarboxylate inhibitor, that's alpha-cyanophosphonate, that it's a monocarboxylate transporter inhibitor. It will inhibit, at the concentration that we used, 0.1 millimolar, it inhibits pyruvate transport into the mitochondria. That this has been our interpretation from mouse data as well. So basically, my interpretation for this is that it doesn't seem to be a difference, an intrinsic difference between central and peripheral rods, but the macula is more sensitive to ischemia than the periphery, as far as we can tell. Fairly weak conclusion, I would say, because it is actually something to be expected. But at least this is what we have. We got something at least, but not as strong and as I would have liked. Okay, so conclusions from the humans: NADPH fraction is about 20%, and the capacity, the NADPH generating capacity of human rods is resistant to prolonged ischemia. So this is just because we get the eyes close to 30 hours after death. So the retina has been ischemic. It does show signs of ischemia when you look at it in terms of how the cells look and so on. And but the capacity, the metabolic capacity for NADPH generation is several times higher than what we would need for retinal to retinol reduction. And just to put things together, what I have been telling you about is a single cell assay for measuring NADPH generation in mouse and human rods. For mouse rods, glutamine and glucose are the preferred substrates. We have not been able to do the experiment in human, and we can discuss why. And for both mouse and human rods, the NADPH fraction is about 20%, which is consistent with Machinsky old results, and consistent with the retinol fraction of about 80%, and which matches our biochemical measurements. And that the generation is human rods are resistant to prolonged ischemia, and that the capacity is several times higher in the rods than what we would expect needed for retinal to retinol reduction. And I'll stop here. I have more things to show if we want to go over the pathways. If there are any questions now, happy to answer, but I'm happy to keep going, but maybe we can talk about some other things.
Okay, we don't have that much time left, but at the
Yes.
same time, I really, really want to see the other data. I want to see the pathways, because obviously
Okay.
glucose is doing more than just, glycolysis
Okay.
and PPP.
Okay. Okay. So, so this is in the mouse actually. So we actually, one of the questions that we had is that, do NADPH and ADP generating pathways compete for metabolic substrate? And the reason is that because they're generated through the same pathways, basically. At least, again, that's my simplistic view from textbook biochemistry or review by reviews in science. So this is actually how I have put the pathway. That's the pentose phosphate pathway here from glycolysis, and then you can actually get the dehydrogenases for mitochondrial linked pathways, and ATP synthase, and you have ATPases that utilize ATP. And so one of the things that we did, we went to 50 micromolar glucose. So from the titrations, we knew that this is mouse. So 50 micromolar glucose is actually limiting. So we actually have, the substrate is actually limiting now. So, okay. And then we're looking, so these ratios are actually down. I have put an NADPH. This is the 340-380 ratio. So so this is the control with 50 micromolar glucose. And what we do is that first we go and inhibit the ATP synthase with oligomycin 5 micromolar. And the ratio goes down, which you would expect. Well, mitochondria cannot make ATP, glycolysis has to make ATP. So the little glucose that we have is actually diverted to make ATP instead of making an NADPH. Okay, so what happens if we inhibit ATPases? And the particular one was the sodium potassium ATPase with wibane, and we get a little bit something extra here without oligomycin. This is in the absence of oligomycin. So this goes on. And we've done this with 50 micromolar glucose or 50 micromolar glutamine, and this is the result. This is 50 micromolar glutamine, 50 micromolar glucose, separate experiments, not together. And basically, this is significant. This was significant. We did not see significance here. They're in general right direction, but basically, what one expects is that what we had expected is that there is competition, as one might expect, that the cell uses the substrate for both NADPH and ATP synthesis. And when you actually limit the substrate, it has to budget which way it's going to go. what's going to use for which metabolite. There's something relevant here, but if we have time, we can chat about it. So the other thing is that with glucose as metabolic substrate, which pathways generate NADPH? So in this case, we are working with 5 millimolar glucose. And what we did actually, 5 millimolar glucose, and we actually take out ATP synthase. So all ATP is going to be made by glycolysis. So this is in the presence of oligomycin. This is what we get. Glycolysis can keep its own, actually. Under these conditions, we have pretty high ratios. And then we go ahead and knock out the pyruvate transporter with 0.1 millimolar alpha-cyano 4-hydroxycinamide. And at higher concentrations, it will inhibit the membrane monocarboxylate transporters as well. But at 0.1 millimolar, seems to be working well. So because we're using glucose as a substrate, do not get pyruvate. So what you get here actually is that you get a reduction. So the interpretation here actually is that it was using mitochondrial linked pathways to make NADPH. And at least face value, maybe there are caveats here, but at least this has been an interpretation. But if that's the case, if we add glutamine, we should be able to get it back. And this actually what happens, bypasses this, and it's at 0.1 millimolar for syn, right? That does not inhibit glutamine transport into the cell. And then we get back the NADPH synthesis. Basically, glutamine actually can provide the substrates actually through the mitochondria, not ATP, right? Cannot be used for ATP, but I can actually make isocitrate and malate to use for NADPH synthesis. So this has been my interpretation of this, that because this is in 5 millimolar glucose, that actually their mitochondria, both the cell is actually using both the pentose phosphate and mitochondrial pathways to generate NADPH. And we can talk about it. I'm being a little bit glib, just a touch. But this is actually the pathways that are relevant. We are out of time. I can talk about my glibness, but
You can talk about what?
about being glib, because basically,
yeah.
The cell actually is managing the ratios, is keeping ATP-ADP ratio at a certain level and an ADPH reduced fraction at a certain level, and budgets itself to try to manage those and maintains a particular flux as needed. I'm talking about generation. It's not exactly generation. It's actually homeostasis, right? It's actually keeping ratios at a particular level. And we're using the ratios as a proxy for fluxes, which is not exactly right.
Yeah, I'm glad you appreciate that.
Yes, I do appreciate that. Yes.
Okay, so people should have the chance to ask questions if they'd like
Yes.
to.
Thank you, Dan.
Sorry for going
in this experiment, I wonder if you're disrupting the NAD/NADH ratios when you inhibit the monocarboxylate transporter, and that's then
yes.
reducing the rate of glycolysis. It could be maybe
that's a good
affecting
point.
your NADPH production.
Yes, no, that's a good point. I know.
And then when you add glutamine, you could be restoring the malate aspartate shuttle via that mechanism.
Yes, yes. No, it's an excellent point. Life is complicated, yes. So we have not tried to actually address that, yes. But mitochondria do many more things than just what I'm putting down up here. But yes, that NADH, we could have checked. We never did. We did not try to look at what NAD, because we can look at NADH fluorescence and see actually if we can pick it up and see if we can actually see from that if we can tell what NADH is doing.
And have you tried a mitochondrial uncoupler to see if you...
Not for this. Yeah, no, we did not. No, we did not. Yes.
Anyone else? I have a bajillion questions. Well, maybe like a few, but So I can ask one unless somebody else wants to. I'll just ask. So I really love the kinetic curves that you have for, glucose concentration as a function of NADPH generation, because I think kinetics can tell you a lot. And certainly, so do you know, for example, the kinetics of the glucose 6-phosphate dehydrogenase, the first, which is supposed to, I think, be rate limiting for the PPP, right? And do you know if its kinetics for, say, glucose 6-phosphate concentration are matching up with how you're titrating glucose compared to like a mitochondrial enzyme?
I understand. No idea. There's so much where the single cell assay can be pushed, and in a sense, the cell is a black box. We give it things, it does things inside. Whatever I have put here actually is what from biochemistry, and of course, I have simplified it enormously, as was been very nicely pointed out. The mitochondria do a whole bunch of other things that will affect my interpretation, the interpretation here. This is, but yes, so there are many ways that things can be done. The only thing I can say is that we have data that could be interpreted this way. Of course, I trust the data. The interpretation, I think it's open, and I think that's just other possibilities could be the enzymes, it could be NADH levels.
Yeah. Well, on that sort of similar note, well, maybe not. So you talked about these human experiments where you had basically been working in ischemic tissue, which I'm
Yes.
amazed you're getting these really neat biochemical measurements from, so long postmortem tissue. I think it's fantastic. But are you concerned at all about, perhaps a change in the, say, number of mitochondria that might happen after death? Is that something you've ever tried, say, in a mouse retina? Does postmortem interval really change, the
Yes.
extent to which you might have G6, pentose phosphate,
Understand.
just capacity versus mitochondrial capacity?
So yeah, no, this is an excellent point. So we tried some experiments with mouse, and let's say, okay, keep the eyes for a few hours or a day, and then check what's going on. And we do not try very hard because we do not get any cells. And but this is anecdotal, right? It says we never tried hard to see if I actually put a mouse eye under ischemic conditions for a day, will I get anything? But the little that we tried was actually very disappointing. So we stopped. We did not try to do it, let's say, go for an hour, 2 hours, 5 hours. We never tried that. What I can tell you is that when we have looked with fluorescence, with mouse cells, and exciting with physioptics. In physioptics, you're going to pick up FAD. And you can, in the mouse, you do not see FAD. So the cells seem to be very nice, working very well. You saw NADH, you can actually look with physioptics and to see FAD. You do not see FAD. In the human, we always see FAD. So my interpretation, again, we never tried actually to see, can we quantify this? It's quantifiable, the redox imaging, right? And but my feeling has been that the human cells have been oxidized, really, to put it bluntly, that really they're bunged up, so they have been stressed out. And it could well be that the human cells could have more intracellular stores than when we get to see, simply because of the ischemic period that have been exhausted. We tried a few experiments with monkey, and this is where the tricky part comes in, because with monkey tissue we could actually get from a local company, and it's within 3-4 hours post-mortem we could actually get cells, and we never were able to tell. There is a possibility that there are actually large intracellular stores, metabolite stores in primates. This is known in cones for glycogen, but it might also be the case for rods. And I do not know what it is, but it's something to be tested. we never went actually to try very hard again with primate tissue. You try to do the experiments that you need, and you need to make sure that you have washed glucose away. And this is the tricky part, right? In the mouse, we could actually try very well. We figured it out, and we knew that we had really no glucose. With primate tissue, we never were able to do it the right way. So I was confident that we did not have glucose around. But then we went on. We wanted to do other things, basically.
Yeah.
With extracellular retinal, that's not a problem. It's just it really overwhelms things. And but we never went around actually. Okay, if we have your regular glucose and endogenous or a little bit, can you tell what's going on? And we could not. So my answer is I do not know, but there is a very clear possibility that there is a significant difference between primate and mouse.
Okay. Well, one more question since you mentioned cone glycogen already. Have you done cones and what do they look like? I mean, are they resilient to adding more glucose because glycogen is enough, or what? What's going on there?
No, we haven't done titration. So the cone experiments that we have done, we get NADPH ratio is about 20%. This is published, and we get the same thing with human and with monkey. And so I'm comfortable with cones working with 20% at 20% NADPH. The reduction now, and that 20%, unfortunately, is the same for frog, I'm afraid, at room temperature. We never tried these measurements in frog, but the retinal to retinol conversion actually seems to be about at 80%, at 80% in the frog as well. So all species actually, at least frog, mouse, human, monkey, convert 80% of the released chromophore to retinol, and that would correspond to an NADPH of about 20%. It's similar things in the frog, and the cellular pH is about the same under these conditions. So cones are the same, around 20% NADPH. I would imagine. We haven't measured pH in cones, but if it's similar in that range, 7.1, I would expect a 20% NADPH. There are many experiments to do in cones, very difficult to do with human. We cannot get really enough cones from the macula. Could not do that. Very, very few. Enough to say that it's 20% NADPH, but that's about it. No other experiments. We had better luck with monkey, but again, we never went ahead to actually try to figure out, is this, are there intercellular stores? Because that has been a question in the back of my mind. Again, we got sidetracked.
Yeah. Does anyone else have any questions?
Dan, I have two. Can you hear me?
Yes.
Yes.
Okay, so I have two last questions. One's maybe for both you guys, and one's for Yiannis. Yiannis, does the setup work for plugging in a genetic mouse model? Like, for example, the MPC knockout in photoreceptors has been shown to have a phenotype, right? And so does that get you somewhere towards isolating from those types of
yes.
Those types of rods, the question of contributions of different NADPH
Yes.
stores?
So yes, we have done different knockout animals, genetically modified animals, never metabolic ones with metabolic pathways. But yes, it does work with transgenics as well.
So maybe, I mean, that
knockouts.
could be interesting, because that obviously
Yes.
keeps
yes.
pyruvate away.
Yes.
And then the second question, just because I'm an RPE person, can you and or Dan or others remind me, like in standard flux experiments, when you label glucose and it's going into photoreceptors, what percent is thought to be shunted into pentose phosphate, versus end up in TCA cycle intermediates?
I'll defer to people more knowledgeable than me.
Less than 1 in my experience for a mouse retina.
Less than 1% is shunted into pentose phosphate?
In my limited experience, they have this, basically it's an isotopic tracer you can take almost ratiometric measurements from. I can barely see a signal that would reflect pentose phosphate, and it has to be that those pentose phosphate carbons re-enter glycolysis.
Right. This is
And
that
it
trick
could be that they're directed away, in
yeah,
which
to
case I
nucleotide
wouldn't
or
see
something.
much.
Got it. Okay.
Any other questions? If not, thank you so much. I thought this talk was wonderful. And
It's a pleasure.
yeah, I'm really happy that you came. And thanks all of you for sticking around.
Thank you, Yiannis.
Thank you. Pleasure.